Swayam Prabha. Digital India. Educated India.
Hello and welcome to today's lecture of NPTEL course Introduction to Mechanobiology. So, in the last class I had discussed about force velocity relationships of actin networks. Okay. So, the advantage of having a branch, so if I draw a cell, okay. So, this is your nucleus. Okay. The polarization of the cell is dictated by the microtubule cytoskeleton, but you have your actin filaments which are forming various kind of structures. Okay. Okay. So, these structures are your actin stress fibers. These long pointed extensions that you see are called philopodia, and this branched network, okay, this portion of the network is lamellipodia. Okay. So, philopodia, so the way the actin is cross linked by various proteins is different in case of philopodia versus in case of lamellipodia. In lamellipodia, the actin is more branched. Okay. It is a branched network versus in philopodia, the actin is more bundled. Okay. So, actin plays a key role in cell migration. Okay. So, actin plays a key role in cell migration. So, what we are going to discuss today is to study how actin participates in directing migration of cells. So, for that let us first look at the various steps which are involved in cell migration. Okay. So, in cell migration, so if I were to draw in side view, okay, let me draw the substrate. This is your substrate. Okay. Your cell is anchored by multiple adhesions. Okay. As a first step, what you have is actin protrusion. Okay. So, at the leading edge of the cell, so this is the leading edge. Okay. First thing which happens is you have elongation of the cell. Okay. So, this remains the substrate. I am not drawing the entire cell. You have actin protrusion. Okay. In the second step, okay, what you see is the formation. So, for the second step, I have to draw the entire cell. Okay. Once again, okay, this is your protrusion. Okay. So, earlier you had focal adhesions here, here, here. What you also have is formation of an adhesion here. Okay. New adhesion is formed. So, first you have protrusion, then you have adhesion. So, what adhesion does, it stabilizes this protrusion, okay? And subsequently in steps 3 and 4. So, in step 3 you have translocation of the cell body, and finally in step 4 you have de-adhesion. Okay. So, what is de-adhesion? I will draw de-adhesion for you. Okay. Okay. So, this adhesion gets broken. Adhesion at the trailing edge. So, trailing is this is the trailing edge of the cell. Gets broken, okay, and as a consequence the cell moves forward. Okay. Now, so the question for today is, can we study how actin polymerization contributes to cell migration. Okay. So, it is obvious that if you have a filament against a wall, so what is the wall in case of a moving cell? The wall is nothing, but this is nothing but the cell membrane itself. Okay. So, because of some tension in the cell membrane, so tension of the membrane dictates the force the filaments are subjected to. Okay. So, we know from in optical microscopy, right. So, I had introduced this term called diffraction limited. Okay. So, it is impossible to see a single monomer under an optical microscope. So, you cannot see objects which are less than 250 nanometers. Okay. So, if I were to draw a filament, okay. So, imagine I have drawn an actin filament. Okay. So, this is an actin filament, okay, and a monomer. So, let's say this distance, okay, is 250 nanometers. So, you cannot see any event, let's say like a monomer going in this direction or a monomer being added here. These events are impossible to see. But if you label the filament, if you label the entire filament, okay, so then this entire thing will turn red. In this case also you cannot see individual monomer level polymerization or depolymerization events. Okay. So, how can we devise an approach such that we are able to visualize protein dynamics? So, you want to visualize okay. So, the approach, one of the approaches developed is called fluorescence speckle microscopy or FSM. Okay. So, what this technique does, so let us assume in the simple case you have a filament, or even let us say this be a microtubule which is entirely labeled. Okay. So, in that case you are in no way to track the dynamics. Okay. But let us say you do the following experiment, okay, in which you take unlabeled filaments. Okay. So, this is an unlabeled microtubule, okay, and what you add is among the, so you have all these subunits which are unlabeled, okay, and within this mix of unlabeled subunits you add some labeled subunits. Okay. So, this is a small percentage. Okay. So, let us say if in this case, if the percentage of labeling was 10%, okay, so what you will see, okay, you would see these roughly the labeled zones interspaced with black zones which are not visualized. But if you reduce the concentration of labeling significantly, then what you will find, okay. So, this is let us say order 1% labeling. So, now you do not see the entire structure, but you can guess what that structure is. Okay. So, you can see multiple of these dots. Now, what do these dots represent? Okay. So, these dots are called speckles, okay, where, okay, so fluorescently labeled subunits have clustered together. Okay. So, in order to image these speckles, the duration of association, okay. So, for imaging speckles, okay, your duration of association, okay, of the subunits must be equal or larger than the time required for capturing a frame. Okay. So, what is the advantage of having of these fluorescent speckle microscopy? Okay. So, I have drawn this particular filament, it might be an actin filament or a microtubule filament, which is, so I have intentionally drawn dots of various sizes, because the chance or the size of these speckles will vary, because this association is completely random. But once you see this speckle geometry, what you can track, there are two pieces of information that you can determine from this. Okay. So, you can track each speckle and you can track speckle movement. Okay. You can track speckle movement and you can track speckle size. Okay. So, if locally at, let us say if this particular speckle is growing in size, then I know that there is some polymerization event going on. Okay. So, increase in speckle dimensions is linked to polymerization events. Okay. So, in essence speckle size is correlated to turnover. Okay. So, if we take, let us say, so if we take a cell, let us say, which is stained, okay. Imagine this is one stress fiber network, okay. This is portion of a cell where you see stress fibers in this cell. If the same cell, okay, were imaged in speckle microscopy, then what you would see, okay, though you will not see the entire structure, but you can gauge the orientation of these filaments from the speckle microscopy. Okay. So, similarly, if your adhesions, let us say for this cell you had these adhesions which were like, okay, in speckle microscopy, okay. So, what you would have found was, okay. So, you would still see, get an idea of where the adhesions are, okay. But not just that, in addition to the static information, now using speckle microscopy you have access to dynamic information. Okay. So, now let us say you use speckle microscopy to image actin dynamics in migrating cells. Okay. You want to ask what is the nature of actin dynamics in a migrating cell. Okay. So, if you have this cell, let us say a section, you have this whole cell, you can zoom into one corner, okay. You can zoom into one corner, and if you look at the speckle images, you would see tons of dots, okay, tons of these dotted particles, okay, or speckles. So, in order to gain any quantitative information, you need to develop algorithms to track spatial position and intensity fluctuation of each and every speckle. So, this is no mean task. Okay. So, there are authors. So, Gordon's Denuser is a mathematician who has significantly contributed to our development of fluorescent speckle microscopy. Okay. So, if you do, now the question is, if you do this speckle microscopy, what is the nature of the movement of the speckles? So, what has been found was, so let us say that this is a cell, you are looking at this edge of the cell. So, what I will do is, instead of plotting the speckles, I will plot what is the overall motion of the speckles. So, what has been observed is, so this edge, let me draw it a little bigger. So, there are actually okay. Okay. So, what I have drawn here, okay, is the distribution of speckle movement. Okay. So, what you are looking at is speckle movement in a migrating cell. Okay. So, this is the direction, this represents roughly, this represents the direction the cell is moving at this point of time. So, what you see that there are two populations of speckles, okay, or two zones of speckle behavior or movement. Okay. There is this zone at the periphery, okay, zone 1, speckles are fast moving, and zone 2, speckles are slow moving. Okay. So, but consistency is all the speckles, okay, tend to move inward. So, if the cell is trying to move outward in this direction, the speckles are moving inward. Okay. So, this motion, inward flow, is referred to as retrograde, okay, actin flow. Okay. So, you have retrograde actin flow, all the speckles tend to move inward, that is why retrograde or backward. Okay. So, backward compared to the direction in which the cells are moving, and you have fast moving speckles right at the periphery of the cell. So, this width of the region where the speckles are fast moving is approximately 1 to 2 micron in width. Okay. So, 1 to 2 microns in width at the periphery, and then there are slow moving speckles towards the center of the cell. Second point, what they found, okay. So, you have two regions of retrograde flow. Okay. What you can also do is, for the cell, okay, you can track, okay. So, you can track the intensity fluctuations, okay, to find out, okay, to find out how does the intensity fluctuation change along the length. So, what was observed, okay, and then okay. Once again, so what I have done, red corresponds to intensity increase. In other words, okay, it signifies a polymerization event. And green corresponds to a depolymerization event. Okay. So, once again, this is the direction in which the cells are moving. You have this. So, again, based on this, I can segregate the entire cell into two zones. Okay. The zone at the cell periphery, where the speckles were exhibited fast retrograde flow, had a layer of speckles where there was indicative of polymerization, and right next to it a layer of depolymerization. Okay. And then inside, within the slow speckle zone, you have mixture of both red and green dots. So, polymerization and depolymerization both. Okay. So, what the authors did was call this zone, okay, of, so zone of high polymerization and depolymerization, and fast retrograde flow. So, this is the zone which they refer as lamellipodia. Okay. And this zone, internal zone, is called the lamella. So, what is the lamella? It is a zone of slower flow and punctate or mixed turnover pattern. Okay. So, you have two zones which seem to be physically distinct. Okay. The lamellipodia which has speckles fast, so fast retrograde flow, high turnover, okay. And second is zone of slow flow and punctate turnover pattern. Okay. So, this suggests that you have these two zones which are right next to each other, but probably they are physically distinct zones. Okay. So, what they went on to see was to check that what is the spatial localization of some of the key proteins, actin associated proteins. Okay. And what they found, okay, was ARP23, okay, which is associated with branching of actin. It is present in the lamellipodia. Okay. Similarly, cofilin is an actin depolymerizing, so it is a capping protein, contributes to actin depolymerization. Okay. This is also present in the lamellipodia. In contrast, okay, myosin 2, which actually pulls on actin fibers, it was enriched in lamella, and same was tropomyosin. Okay. This was also present in lamella. So, in short, what you have is two zones which exhibit distinct retrograde flow, distinct turnover, and also are molecularly distinct. Okay. So, suggesting that there must be some dynamics which is different in these two zones. Okay. With that, I stop my lecture today and will continue from here the next day. Thank you.